In this short video, I'll talk about the mechanisms related to epithelial barrier theory. And we are all wondering why these diseases are developing. And the mechanisms can be summarized as epithelitis and expulsion response. So there is a homeostatic and healthy interaction of microbiome and epithelial cells. And the epithelial cells are very tightly bound to each other. And even water cannot pass through them. And there is some immune system cells below the epithelial cells that get some signals from the microbiome. Dendritic cells send their dendrites and recognize the environment and also get signals from microbiome. There are B cells that develop IgA type of antibodies. And some very small numbers of innate lymphoid cells and T cells are in the tissues that are in contact with the epithelium. But when this interaction is broken, when epithelial barriers are open with the substances that we have mentioned, with the toxic substances like the cleaning material, sodium lauryl sulfate, and the shampoo and toothpaste, and packaged food emulsifiers, and additives in packaged food, and also air pollution and many aspects of air pollution, particulate matter, cigarette smoke, ozone, microplastic. This epithelial barrier is broken. Then the first thing happens is that the microbes go deeper in these epithelium and then they translocate under the epithelium. So we have a translocation of microbiome below the epithelium, and then there is colonization of the opportunistic pathogens for the respiratory area, for example. Staph aureus, Moraxella, Haemophilus, Pneumococcus are colonizing. And then they make a danger, actually, because these bacteria can go to deeper tissues and then they can cause abscess in the tissues, or they can make bacteremia and sepsis, or they can go to far organs and make brain abscess, lung abscess. So it's a big danger now that's happening here. And the epithelium first responds to this when it is contacted by the bacteria on the sides of the epithelium or beneath the epithelium responds by releasing of allergens. There is inflammasome activation and the release of IL-1 beta, then alarmins IL-25, IL-33, TSLP, and multiple chemokines are released to initiate an inflammation. So epithelium is inviting the immune system to the area and saying that, look, my barrier is broken and some dangerous things are passing the barrier, so come and help so that these bacteria do not cause sepsis and they do not cause a severe disease. And immune system comes to the area to localize these bacteria and trying to throw it away. As the next step after localization of the immune system, there is a chronic T and B cell response to invading agents. There is allergens, microbiome, opportunistic pathogens, and autoantigens are all receiving a T and B cell response because the immune system doesn't see the difference between the microbe or autoantigen as at the single antigenic epitope level. And this total immune response starts an expulsion response to microbes, allergens, toxins, and pollutants that are invading the tissues, that are going deeper in the tissues. And another important reason is that the immune system is there to avoid sepsis, because Staph aureus is there, Haemophilus, Neisseria, Pneumococcus, they are all dangerous agents to cause sepsis, and the immune system responds to this. And then there is, in the first step, there is major players are IgE, eosinophilia, open barriers. There is cough or itch reaction, Th2 cells, B cells, type 2 innate lymphoid cells, and with their major cytokines, IL-4, IL-5, IL-13, are playing a role in this expulsion response. And with the tissue pressure, tissues are pushing all of these invading agents to outside with the inner tissue pressure in the range of 10 to 15 centimeter water. And then at the same time, during this inflammation, when we carefully look at, we can find that there is a detectable autoimmune response to skin keratinocyte antigens, to the lung antigens, as demonstrated by Rudolf Valenta, Reto Cremeri many years ago. and the neuroimmune interaction is also taking place to set this inflammation. When we look at this whole picture, it is very similar in chronic rhinosinusitis, allergic rhinitis, asthma, and atopic dermatitis. So different tissues show a similar response with the colonization of opportunistic pathogens and an immune response trying to throw them away. And at the same time, we have allergen-specific immune response. We detect allergy, we detect IgE against allergens, but the total immune response is not only against allergens, it is against the microbiome, and it's against the opportunistic pathogens, and also many times against autoantigens. So in summary, in epithelial barrier defective tissue, we have exposure to barrier damaging toxic substances, and with genetical reasons, a barrier can be open, and there's colonization of opportunistic pathogens, translocation of microbiota to subepithelial areas, immune response to allergens, to commensals and opportunistic pathogens, and to autoantigens is taking place, and microbial dysbiosis happens. So the microbiome changes its characters because of the immune response, because of the effect of toxic substances altogether. And there is periepithelial inflammation and chronicity, and defective barrier healing capacity of the epithelium, and due to chronicity, this reaction continues, and this causes 2 billion allergic, autoimmune, and neurodegenerative patients. So in summary, we have the direct toxicity of the epithelium damaging agents, then epithelitis and microbial dysbiosis takes place, and then expulsion response is taking place. And there is also a circulating inflammation is playing a role in bringing this inflammation to other far tissues. Like if the gut is inflamed, the diabetes gets worse, or fatty liver gets worse. And these kind of far organ interaction and gut-brain axis and gut-lung axis, gut-skin axis, they're all playing a role, or lung-brain axis are playing a role to make the chronic diseases. And epigenetics and changing the characteristics of the epithelial cells to heal or to recover is also taking place. We have seen years ago when we biopsy asthmatic epithelium and when we culture the asthmatic epithelium in organoids and in organochips or in air liquid interfaces, asthmatic epithelium can never make a full strong barrier. And this is known for almost 15 years. And here we have the whole picture of the epithelial barrier theory, starting from the epithelial barrier damage and microbial dysbiosis and immune response against many agents that are invading the body, like the microbes, but also an allergen-specific strong immune response is taking place at the same time. I stop here by thanking our co-workers.